Alright, so this will be our review for exam number 1 for VITI 301, Blood Break to Harvest. It'll be covering lectures 1 through 4, and we're actually going to hold this exam, oddly enough, on Friday. You can take it anytime between 8 a.m. to 11:59, right before midnight. So if you have any problems with that, just send me an email. Normally we would have our exams on Wednesdays, but I wanted to give you guys a couple extra days just so you could be prepared for that. But if that conflicts with your schedule and you want to take it Wednesday, just let me know. Okay, so chapter 1 was grapevine anatomy and physiology. So what we'll do is, if you haven't been to these review sessions before, is we go through the lectures and we just kind of go over some of like the important topics just to kind of review together. I do oftentimes take questions from these review sessions, so definitely benefits you to watch and review with me during these sessions. I do upload them to YouTube, so you can watch them multiple times if you'd like to, or if you couldn't make the Zoom meeting, you can catch up on your own time there. I am recovering from a cold, so if I start coughing, I apologize. I do have a cup of tea at the ready. So let's get started. So anatomy and physiology, we kind of broke down the difference between cells, tissues, xylem and phloem, the epidermis, the stomata, lots of vocabulary in this section, and lots of memorization. So any words in bold, right? Just like our quizzes. The exam is going to be just like our quiz, but it's going to be longer. So it's going to be 100 points and multiple choice, matching, short answer, true or false. Very much just like the quizzes, but just longer in length, right? Okay, so we have plant cells. Plant cells have a cell wall. That's what makes them, specifically different to animal cells, right? And unfortunately, my video is in the way here. Let me adjust that really quick. Just make me a little bit smaller. There we go. Okay. There you go. Okay. Cool. So like I said, lots of vocabulary in this section. Definitely know that plant cells have a cell wall, right? They also have a vacuole where they have cell sap stored. I think for a question that for a lot of these vocabulary words, it's going to be mostly matching. I don't think I'm going to ask you to come up with a definition on your own. So if you can recognize the word and recognize the definition that's correct for that, I think you'll be in a really good spot for this exam. And I might even potentially, I'm not going to do that. I was going to say I might have you match to the picture, but we didn't do that in the quiz, so I don't feel like doing that to you guys. Okay, for tissues, again, a lot of vocab in this one. The cambium layer is very important because this is what we talk about in grafting for VITI 303 in the fall. This is the part that makes it determine whether or not it's successful for grafting median to take place. So there will be some focus on the cambium layer. Definitely be able to tell me why it's important and where it is in the plant, right? Cool. And we did do a lot of focus on the difference between xylem and phloem. This was a quiz question for you guys, so I would be prepared again to be able to compare and contrast the two of these structures. And you guys did a really good job with this on your quiz. So if you would like to study your quizzes but in preparation for the exam, they should be open for you. If they're not, send me an email and I'll fix that right away, but that's a really good tool to study. Also, the student learning objectives at the end of the presentation are also a very good tool to prepare yourself for the exam. Okay. And just another representation of the xylem and phloem. Okay. Tissues. Epidermis, this is the skin. You have cutin and stomata. Lots of focus on stomata, gas exchange, and water regulation. Whenever there's like a numbering, like a 1 and 2, I always like to put that in a quiz. So be prepared to talk about stomata and the two things that it regulates, right? Okay, grapevine anatomy. This would probably be a picture where you have to identify the different parts of the vine, so be ready for that. Part 2, again, more anatomy and physiology. Okay, more vocabulary words here. Understanding the process by which water is pulled through the grapevine, right? So it evaporates through the leaves and then it creates a transpiration pull from the roots up to the plant. So be prepared to summarize that, I'd say, in a short couple sentences or small paragraph, and just understand that function. More on the stomata, because I just love stomata, I guess, and how they operate, photosynthesis, osmosis, more vocabulary words. Sorry if it feels like I'm blowing through this. We would just have to expect a lot of vocabulary and on this part of the exam. I'm not going to ask you to write out anything like this. Definitely understand the difference between cell respiration and photosynthesis, right? They're opposites of each other. Be able to recognize that. And then learning objectives. Cool. Any questions on lecture 1? All right. Oh. Hey. Oh, okay, good. Perfect. Thank you. Awesome. Okay, moving into climate for viticulture. Okay. So climate's impact on grapevines, grape development, and also wine style and quality. Some of the factors that we talked about: temperature, rainfall, humidity, and sunshine hours, wind influence. Temperature. This section could have a lot of questions about like throwing hypothetical situations at you. So I could say, you have a vineyard and you're sampling grapes, but you've had a week of extremely hot weather and the fruit is not progressing. What's happening? You could say, due to temperatures, and I would say specifically in the question, like it's been 105 degrees, for 5 days in a row. Then hopefully you'd be able to point out in your response that because of the temperature, the vines are shutting down, right? And then a couple more vocabulary questions here: diurnal temperature variation, the difference between the highest and lowest temperatures of the day versus night, and growing degree days and temperature extremes. Cool. Frost prevention, just being able to identify what prevents frost, right? And why frost is a problem for vineyards. The balance of rainfall and humidity. So just a lot of risks in general to vineyards in this section. The consequences of too much and too little rain. Be prepared to list these potentially on your own. That'd be a good question. Like, what are the consequences of too much or too little rain in a vineyard? And then for knowing if vines have enough water, this could be something that you could recognize potentially in a question as well. Okay, sunshine hours, essential for photosynthesis and sugar accumulation. Will affect acid retention and phenolic development. So again, this could be another hypothetical question of you have a neighbor who has a vineyard and their vines are overgrown and their fruit receives no sun. Why would you recommend that their fruit has sun on it potentially, right? And so you could be able to talk about these things. Okay, wind. Wind is very important. It can damage vines, right? It can damage vines, but it's also if it's too extreme, but it's also important in lower amounts to help take any potential humidity and mildew capability from the vines, right? So there are ways to mitigate wind, and we talk about that. The straw wind was just a really good example of an area that receives extreme wind. But the big takeaway I want you to understand is how vineyards, work with high wind conditions. So planning, orientations, planning windbreaks, fine tuning methods. I might ask you directly in a question how to work around wind, or it might be phrased in some other way where I'd ask you to, but I'd want you to know the different ways. Cool. Awesome. And you guys all did very well with that on the quiz as well. Okay, there will most definitely be a question on what to expect from cool climate versus warm climate wines. So if you're not completely sure about that, definitely reference this chart. This is a really good true or false section material. I like to be really tricky. I like to say something along the lines of wines produced from warmer regions typically have, I would say higher acidity and lower sugar. Well, that would be false, right? Because that's a cooler condition. So be careful when you read my true or false questions, because 90% of it might be correct, but there might be that one word that's not correct. So just make sure you read them thoroughly before answering right away, and you should be good to go. But definitely know the difference, and then also the difference in flavors as well too. So if I asked you to compare a Chardonnay from cool climate versus hot climate, this is a really good chart to reference and can be really helpful on the exam. So definitely know those differences. The Winkler Index might be paired in with a climate question. So I could be a little bit tricky, and I could say, instead of directly saying a cool climate versus hot climate, I could ask you to describe a Chardonnay from the Winkler Index region 1 versus region 5. So 1 being the coolest and 5 being the hottest. So I could tie that in a kind of a tricky way there. Growing degree days, just another vocabulary word for you guys. I will not ask you to memorize any of this. This is just purely for your learning pleasure. But just know that relatively it gets hotter as the regions go up. Cool. And then also understand how the Winkler Index applies to grape growing knowledge, right? If you are going to plant a vineyard, you could compare your region to other regions of the same type and what varieties they plant, and so forth, right? Okay, and this is just a compare and contrast of Oregon versus Central Valley for their Winkler Index, and then the varieties they grow and how the fruit is different. So just learning there. Climate change. You guys did really good on this question on the exam as well. Just know the challenges that are coming from climate change, and be prepared to talk about potentially how wineries are responding to climate change, right? So we talked a little bit about how there could be pros and cons to climate change. So new regions that have never made wine before are now making wine because it's warmer, and that's very positive. Like the United Kingdom, for example, is making amazing sparkling wine now. On the downside, Champagne's having a really hard time because it's much warmer than they'd like it to be there. So you could talk about the pros and cons. And then also another thing that's happening is didn't explicitly say in this presentation, but it might be offered up as an extra credit question, because temperatures are rising in these grape growing zones, people can either move the location of their vineyard northward on the northern equator, or more south on the south of the equator, or they can plant their vineyards at higher elevations, because the higher you move in elevation, every 1,000 feet or whatever, the temperature drops. So it's a cooler growing region the higher up the mountain you move. So that could be a really good extra credit question for those who were paying attention to this video. So take note of that. Okay. Cool, and then we have our learning objectives for lecture number 2. Any questions on that? Okay. On to lecture 3, soil basics. Some more definitions, vocabulary for you guys. Definitely know the difference between field holding capacity, drainage, and then soil texture, right? How soil layers or horizons are made through weathering and time. The different horizons, layers are moved by wind and water. That'll be important. And also the top 2 layers contain the most life in the soil, right? So the organic and the A horizon have the most organic matter and also life, right? It's where all of our animals and organisms live. Okay, not so much on that slide there. The erosion. Okay, yeah, I think we're good. Just a good picture here. Okay, soil composition. It's mostly mineral fragments. There's air in soil, there's water in soil, and there's organic matter as well. That is the whole pie of what soil is composed out of. As far as soil texture goes, there's only 3 types of mineral rock particles, and that is sand, silt, and clay. Sand being the largest, silt being in between, and clay being the very smallest particle. And with these things, we're able to determine the texture classification, which we reference this triangle chart to. There'll definitely be a soil texture question. You'll just have to find the different percentages and locate which soil texture it is. Okay. There could be some questions about how to determine certain things, what type of testing you would do to determine soil. So the jar test helps you determine soil texture and type. It helps to, it separates out the different textures. They settle out depending on their particle size. Then you can determine the percentage based off of the whole sample, and then you can use this chart to determine your soil texture classification. Water holding capacity, it's determined by soil texture and organic matter. This is very important because it'll determine how much irrigation a site will need. So going through these slides, the important things I want you to know is how is this applicable to your vineyard, right? Why is this important to know? And how would you determine this, right? How would you measure this? These are the 2 things I want you to take from all this information. Okay, field capacity is what sticks to the soil after all the excess water has drained out, right? So that's the field capacity. That is what it holds after all the excess is drained. Wilting point is when no more water is available to plants. I think this is a really good comparison of definitions. I might ask you to compare these 2 on the exam. Deeper soils will retain water longer than shallow soils, and soils with high organic matter will retain water for a longer time because it acts like a sponge, right? So this could actually be a good short answer question. I could come up with a question along the lines of vineyard site A has deep soil with high organic matter. Vineyard site B has shallow soil that is mostly just sand. Which vineyard has a higher field capacity and why? And then that could be your answer. Well, organic matter holds more water for a long time, etc. Being able to give a brief description as to your reasoning behind that would be helpful. That would be a good question. Okay. The difference between texture and structure. So you think texture, think of the soil triangle, the chart that we look at. Structure, think about it's the how it holds together, right? So the aggregation of soil particles into larger units. That'd be a good question. What is the difference between texture and structure? So structure is extremely important because it relates to soil health, right? So infiltration and drainage. When soil is highly compacted, it doesn't have any texture, that is when it's very difficult for life to thrive in soil, right? So these aggregates create these like clumps, healthy clumps in soil, and they form through natural forces, organic substances from roots, glomalin produced by fungi, and then also earthworms, other soil animals, right? So this is what makes it available for water to drain and air to be available for roots. Cool. We have a really good photo here showing the difference between healthy soils and non-healthy soils. A bit more, and then again, just to reinforce the point. Okay, water infiltration. Some more vocabulary words here. Let's see, do I want to add anything to this? No, I think we're good. Okay. Bulk density is the measure of soil compaction or lack of porosity. I would just memorize that. No need to memorize this formula. I'm not going to have you do any math for this exam, especially since we did not go over that together in class. So don't worry about that. But do understand why soil compaction is measured, right? Why and also how, right? So why? Because it's going to be very difficult for anything to grow if your soil is too dense, it lacks porosity. And how, right here, okay, is with this little cone. But so it would just be called the bulk density test. Okay. Let's see, anything else I want to add to this? No, okay. Okay, evapotranspiration, another vocabulary word. Cool. So I would like you to know the difference between bulk density and infiltration tests. So understand the difference between these two things. I think that would be a good question. And more student learning objectives. Cool. Any questions so far? Okay. Grapevine dormancy. Okay. So now we're kind of moving into the cyclical part of our class. We covered dormancy and then we shift towards bud break and also harvest. So dormancy, there's the 3 stages, right? Acclimation, midwinter dormancy, and de-acclimation. It's going to be very important to identify those three things, and then also briefly summarize what happens at each stage. So stage 1, acclimation. The vines are acclimating to go into a deep sleep, right? They're pulling in sugar to store for winter reserves. They're going to adjust to daylight and temperature, the big things, and they're going to start to lose their leaves as well. Leaf fall can be a problem if it's too early in the situation. Be ready to talk about that. What causes early leaf fall? That was a quiz question. Okay, identify why starch is important. Solubility, spatial efficiency, and controlled release. Also, what is starch? Starch is comprised of glucose molecules chained together. Okay, I don't think I talked about bud dormancy in the quiz. That's funny, I just wrote that quiz. I should remember this. But I definitely do want you to remember this for the exam. So the two methods, dehydration and supercooling, just be able to outline that, and summarize what those are. Okay, stage 2, midwinter dormancy. Be able to summarize deacclimation. Okay, deacclimation. A problem, right? Because of potential frost damage, right? So if your vines deacclimate, too fast, the buds start to open. This could be a huge problem if a frost comes through, right? So it's been a big problem for us lately in these vineyards. How to determine if your vines have carbohydrate storage, iodine test, or send samples to a lab. Grapevine pruning. It's the balance of grape production and vegetal growth of the vine. So fruit versus vegetal growth, right? Understanding to balance those two things is very important. Determining vigor through the size of the cane is very important too. So you want to be pencil sized. There it is. Aha. Pencil versus like the size of your thumb, right? So just think of John Wick. Great movie. And it's always a balance of the yield, vegetative growth, but also like the quality of your fruit too, right? Cool. So if you have over vigorous shoots, you should leave behind more buds so they're thinner. If they're too skinny, leave behind less buds so that they're more hardy. It's always a good balance. I know I'm kind of going backwards here, but sap flow is a good sign you aren't pruning too early. Know the risks of early versus late, and the difference between overcropping versus undercropping and their consequences. Okay, any questions on that? All right, well, that is lecture 1 through 4 for you. Thank you for attending, and sorry for my coughing attacks. I apologize. If you guys have any questions, feel free to send me an email. There you go.